thank you very much. And that was another fascinating talk. And I'm going to hand swiftly over to the iGEM competition team. I'm not going to say much about it because you guys are going to talk about it.
So our presentation this evening is on African sleeping sickness, or human African trypanosomiasis, HAP for short. There are as much as 60 million people at risk in Africa, which is about the same as the UK population. And this disease is about 100% mortality rate if it's left untreated. Sleeping sickness is caused by a parasite known as a trypanosome. This replicates in the blood and crosses into the brain in late infection, causing the symptoms which the name is derived from. The disease is spread by the tsetse fly, shown on the bottom right, and as with many diseases, all ages are susceptible. The life cycle is two parts, one in the host, the other one in the vector, the fly. Bites from the fly can take in infected blood, which can then be passed on to further hosts in the same way as malaria. This is what's known as a vector-borne disease. Symptoms can take up to 2 years to become apparent after a bite. Diagnosis equipment is currently quite bulky, as can be seen from our example on the slide. After positive diagnosis, a further lumbar puncture is often carried out. This is carried out in hospitals in the UK, as it is classed as a risky procedure. As you can guess, this procedure adds even more equipment that needs to be transported, as well as needing trained staff. Current diagnosis methods are the CAT test, as well as recently released BioLine HAT test. One of the main issues, especially with CAT, is the need for refrigeration, as many diagnosis tools, as well as treatments, need to be kept below 4 degrees Celsius in Central Africa. War in many regions has caused difficulty in the treatment, causing a resurgence in the disease. Adding to this, transporting all the equipment seen across a very difficult terrain has caused even more problems. All this transport often needs to be done quickly to keep diagnostic equipment and treatments cool as well. So what can be done? Development of cures can take years simply to get to trials, and so an early diagnosis is preferable. Once symptoms develop, brain damage has already occurred. So there are some treatments already, but can have side effects later in stages of infection. This is another reason for early diagnosis. One solution is to create a bacteria-based diagnosis tool, as it can be grown continuously on-site and tolerate high temperatures. As well as this, it will be cheaper to produce than normal diagnosis methods. Another great feature is that seeds can be taken from this to create more diagnosis centres elsewhere. So how could this be made? iGEM is a competition run by MIT for the past 10 years. It aims to provide students with experience working in the field of synthetic biology, while also addressing real-world problems. This year, over 3,000 students from over 200 teams have taken part in this competition. Synthetic biology is a broad range of subjects, including genetic engineering. It is generally the process of readapting a feature from nature to carry out a process which it was not initially for. During this summer, our team worked on modifying lab E. coli to detect sleeping sickness.
Okay, so this is our team. You see Anna, Joseph, Konstantin, James, and me, Jamie, and Martina. Our logo is the tsetse fly, the vector responsible for spreading HAT, and it has our university logo incorporated into its thorax, and our slogan is "Wake Up Sleeping Sickness." So the immune system always plays catch-up with HAT, and that's basically because every time the immune system identifies it and launches an immune attack against it, it can switch its surface expression or its disguise in a way. And this means that the immune system is never able to ever fight off HAT. So antibodies are highly specific, sticky molecules produced by our immune system. And they bind to the surface markers on HAT. However, because HAT switches, and you can see here, they've already switched from A and B to C and D, those A and B antibodies will not stick to those C and D surface markers. So unlike, so the way we can detect whether someone's infected with HAT is by expressing the surface markers on a bacteria. and these will bind to all antibodies produced against HAP. They are, in a sense, universal surface markers. False positives are a bit of an issue with current diagnosis kits, and that's why further testing such as the lumbar puncture is often needed. So we designed our assay with what's called an AND switch, which basically means we have dual inputs, and both inputs have to be positive in order to get a final positive output. And this is how it works. So you have bacteria 1 and bacteria 2 that expresses surface marker 1 and will bind to antibody 1 if that's present, and bacteria 2 expresses surface marker 2 and will bind to antibody 2 if it's present. So if both antibodies are present and both bacteria are present, and bacteria 1 sends a signal to bacteria 2, and that releases what's called green fluorescent protein, which is how we know the person is positive for HAP. Because green fluorescent protein is not visible to the naked eye, we needed to have some sort of machine for detecting whether it was there. An off-the-shelf model would cost you about £1,000, but they're extremely accurate and precise. We don't really need that. So we thought we might be able to make one ourselves for a lot less. The way it works is you have your sample that may or may not contain GFP. You then pass ultraviolet light. GFP converts ultraviolet light into green light. You then filter out any residual ultraviolet, filter out any other light that isn't green with another filter, and then you have an LDR which will tell you whether green light is present. If it is, then it's a positive test. So this is what it looks like. As you can see, it's kind of thrown together from bits and bobs we had lying around the lab. We also have an Arduino board and a Raspberry Pi. That's sort of the brains behind it, and it's connected to a laptop just to record the data. This thing set us back only about £30 and could really be done by anyone. And this is the kit we envisaged for the future. As you remember back to that previous slide, with all the bulky equipment that's currently needed to diagnose HAP, this would be a lot smaller. You just have a few very simple things: filters, echinodols. There's your 2 bacterial strains in test tubes that you could keep growing and growing, and a blood sampling kit.